everyone, and thank you for joining. As already introduced by Aisling, I am Jyoti, Application Specialist from Technobis Crystallization Systems, and I am here to talk about enabling small-scale analysis during crystallization screening through a single platform. So the presentation will be 30 to 35 minutes, and after that, we will have a question and answer session. So please write your questions in the chat box, and I will try to answer all of them in the end. Can you go to the next slide, Irina, please? Yes. During this webinar, I will give a brief introduction about the Technobis Crystallization Systems. To give a background introduction to the topic, we will discuss the challenges in the crystallization, and then the important factors in crystallization development like solubility, nucleation, polymorphism, particle shape, particle size, and particle size distribution. We will go through each factor and parameters and discuss why it is important to measure these properties and how we can measure them using these small-scale single platforms, and then conclusions and Q&A session. Next slide, please. About Technobis. Technobis Crystallization Systems provide instruments for accelerating crystallization research. Next slide, please. The product line of Technobis consists of 3 multi-reactor crystallizers with application at different stages of solid-state research and development. Arina, can we go to the next slide, please? And can just click on the new, all the slides. Yeah. So first is a crystal breeder for the early stage discovery with unique features of different crystallization modes, like cooling, evaporation, and vapor diffusion setup. It has been used by many researchers specifically for single crystal growth, along with other applications like solubility and solvent screening. But for solubility and solvent screening, we have Crystal 16 is the pioneer instrument with transmissivity technology. So the latest version of Crystal 16 has integrated analytical software for nucleation rate analysis, which we discuss in detail. Then the Crystalline instrument for the optimization of the crystallization process, where different crystallization modes or different crystallization methods like cooling, evaporation, seeding, anti-solvent crystallization experiments can be performed easily. And during the experiments, various operating parameters such as stirring type, stirring speed, temperature profile, volume, etc., can be varied easily and optimized. The Crystalline combines the temperature and the turbidity measurement with the real-time particle image capturing, and therefore it has unique application in crystal habit control, particle size distribution, polymorphism, and process optimization. So all 3 instruments work on different volume with different number of reactors. Next slide, Ina, please. So now coming to the topic, why do we need a small-scale analysis in crystallization? First, we define crystallization. As we all know, crystallization is a separation and purification step used across various industries such as pharmaceuticals, chemical, food processing, etc. Although in simple words, crystallization defined as ordering of molecule in a structured form, but is highly complex process because there are different factors that influence the crystallization and the final product quality. First is the chemistry and the compound themselves, and then the choice of the solvent systems and any additives used or any anti-solvents used. So these chemical choices can drastically affect the crystallization outcomes. Then thermodynamics, the temperature, thermodynamic parameters like temperature, solubility, supersaturation, they play an important role in crystallization process. Then process parameters, the scale of the experiment, mixing, which include stirring type, stirring speed, order of the operations in the procedure like heating and cooling profile, then material of the vessel, etc. So there are so many different factors which affect the crystallization process. Now, addressing these factors become challenging and requires understanding and controlling the crystallization process. Arina, can you click next, please? Next to twice you can click. Yeah, one more time. Yes, to understand the crystallization process, which requires, can we go back, please? Requires a large number of experiments, and these experiments needs to be accurate and robust, meaning they need to be replicated and verified under different conditions. And importantly, in today's cost-conscious environment, these experiments needs to be cheap and easy to perform. Next slide, please, Arina. Yes, so this brings us to the importance of small-scale analysis during crystallization screening. So these single platform facilitates rapid screening and precise control, making crystallization research more accessible and less cumbersome. So Crystal 16 with 0.5 to 1 ml of working volume with the 16 reactors, and Crystalline with 2.5 to 5 ml working volume and 8 reactors. The facility of multiple reactors and small volume adds advantage to these instruments than any conventional crystallizers. Arina, can you keep clicking on these? Next. Yes, like small amount of material needed, inline analytics, no cleaning required, cheap disposal of the material, and little training required. Next, Arina, please. So there are several important factors that affect the crystallization development as listed here. Solubility, which is the foundation of any crystallization process. Nucleation, which is the first step of crystallization and has a significant effect on the final crystal attributes. Then polymorphism, it defines the purity of an API. And finally, it is important to control the particle shape and particle size distribution, which has significant influence on the downstream processes and the end application. We will go through each parameter and discuss why it is important to measure them and how we can measure them using these small-scale single platforms. Next. First is the solubility. Next. So solubility is defined as the amount of compound that can be dissolved in a given solvent at specific temperature and pressure. So it is fundamental information that is required to start any crystallization process. And solubility data are used to take critical decisions from the earliest stage of drug discovery throughout the entire process and the development and up to the formulation. Next. So how do we measure solubility? How do we measure the solubility? This is a phase diagram representing temperature-dependent solubility and metastable zone width here, which gives a lot of important information. The blue line, it represents the solubility curve and defines the equilibrium concentration that can be dissolved at specific temperature. Next, anything below the solubility curve is a solution that is in one phase, and this red line is the metastable zone curve and defines the start of the nucleation. And anything above the metastable curve is the suspension, and this is in two phase. The region between the two is the metastable zone width, where the crystal growth takes place, and this metastable zone width is also your seeding window, where you can provide the seeds to start the nucleation. Further, from the solubility curve, you can determine the theoretical yield from the solubility at the starting temperature and the solubility at the end temperature. Next, how do we measure the solubility? So there are three different methods to determine the solubility. First is the gravimetric method, or also known as equilibrium concentration method, in which you add the known amount of compound or material in a solvent at a given temperature and keep it stirring for a long duration. After that, you filter the solution, and from the undissolved compound, you can determine the solubility. This method is considered to be most appropriate, but it is very time consuming. For one solubility curve, it can take few days. Then the second is the solvent addition method, in which you add solvent dropwise. And when all the compound is dissolved, it gives you the solubility. So this method is also very time consuming, and it may not give you accurate concentrations. Then the third method is the polythermal method, in which the temperature keeps changing, or the temperature variation method. To measure solubility by this method, you add known amount of compound or material in a solvent and keep increasing the temperature. The temperature where all this compound is dissolved will give you the solubility values. Next. So Crystal 16 and Crystalline uses the temperature variation method because it gives the reproducible results fairly quickly, and also determines the metastable zone width, which is not possible by any other method. Then you can have 4 solubility curve in just 4 hours. Next. So how it works? It works on the transmissivity technology. The light is transmitted through the sample, and based on how much light is scattered, turbidity is measured. So the instrument measures the change in the transmissivity with the change in the temperature, which determines the clear and the cloud points. So the clear point is the temperature where the first 100% transmissivity is obtained, indicating the complete dissolution of the solute. While cloud points is defined as the temperature upon cooling, where last 100% transmissivity or first 0% transmissivity is obtained, which signals the appearance of the crystals. And the difference between the clear and the cloud points gives the metastable zone width. From the clear and cloud point, you can determine the solubility curve and metastable zone width curve. So there are numerous research available to determine the solubility using Crystal 16. Next. So there is another method to determine the solubility by capturing the clear and cloud point from the images taken using the crystalline instruments. So this is very useful method in determining the solubility for colored compounds, where the transmissivity is affected by the color, and the images gives you more accurate clear and cloud points. You can select the clear point, where you can see the last crystals while increasing the temperature, and cloud point, where the crystal starts to appear again in the images while decreasing the temperature. You can also define your clear and cloud point on the basis of the particle detected or number of particles detected, represented by the blue curve in the graph. Next, so this is an example of solubility and the metastable zone curve determined from the images using the crystalline instrument. Next. Coming to next is the nucleation. Next. So nucleation is the first step of crystallization, in which phase transitions occur from supersaturated liquid phase to a solid phase, and the nucleation has significant influence on the next step, that is the growth phase, and subsequently on final crystal attributes like polymorphs, particle size, particle shape, and particle size distribution. Therefore, it is important to determine the nucleation kinetics. Next, now how to determine or measure the nucleation kinetics. Nucleation is a stochastic process, and therefore it is difficult to know the actual mechanism for nucleation, but there are different theories, mainly 2 different theories, which propose or explain the mechanism, that is classical nucleation theory and non-classical nucleation theory. So the classical nucleation theory is commonly used and well accepted method to determine the nucleation kinetic of small molecules. According to classical nucleation theory, the nucleation behavior of a solute in a particular solvent originates from the energy barrier for nucleation. So this equation is the energy change associated with primary nucleation, where the molecules combine. When the molecules combine, they form the cluster, and if the size of the cluster is more than the critical radius, represented by C critic here, it forms a nucleus, otherwise it dissolves again. Next. Then according to classical nucleation theory, the nucleation rate J depends upon the nucleation ratio S following this equation, following the given equation, where A is the kinetic parameter and B is the thermodynamic parameter. So one of the methods to determine the nucleation kinetic experimentally is by measuring the induction time. Next. So crystal 16 can be used to determine the nucleation kinetics by measuring the induction time. For that, first we prepare the 4 different samples with different supersaturation using this formula S is equals to Ci upon Cs, where Ci is any concentration that you want to prepare, and Cs is the solubility at temperature where you want to keep your samples constant. Then you program your experiment by first increasing the temperature and maintain at a higher temperature until everything is dissolved, which is represented by the clear point. Then crash cool at a very fast cooling rate, for example, 20 degrees Celsius per minute to some constant temperature, for example, 20 degrees Celsius, and maintain at the same temperature for long duration. The time between the formation of supersaturation, represented by a small green mark, and the appearance of the first crystals, represented by the cloud point, is called or calculated as induction time. Then repeat the same experiments for more than 80 times at each supersaturation. Then from the large number of experiments, the Crystal 16 software plots the probability plots from which gives the nucleation rate and the growth time. Next, then further using the nucleation rate and the growth time values at different supersaturation, the software plots the classical nucleation equation as a linear plot, and from the intercept and the slope, the nucleation parameters A, that is a kinetic parameter, and B, thermodynamic parameters, are determined. So this gives the nucleation rate of a compound in specific solvent, and all these calculations are done by the Crystal 16 software. There are no manual calculations. Next, next is the polymorphism. So what is polymorphism? In simple words, polymorphism is the ability of a material or compound to exist in more than one form or crystal structure. So these are the different arrangements how molecules can be arranged. For example, amorphous, in which there is no particular order of molecules. Then form 1 and form 2, where molecules are arranged in different structures. And if we incorporate secondary molecule, where in which, for example, with different solvents form solvates, and with water form hydrates. And then if there is ionic bonding, then we get the salts. And if there is hydrogen bonding, then we get the co-crystals. Next, but the important question is why it matters. So it can be explained in 2 factors: biological factor, which includes the solubility, dissolution rate, safety, dosages, etc. So that means what effect it has in our body when we take the drugs, because different polymorphs can have different effects, and the dosage will change with the change in the polymorph. And then the economic factors, for example, density, flowability, drying, and packaging properties. That is how easily the API can be packed into a tablet. Some polymorphic forms with different crystal shapes can be difficult to form into a tablet and can cost a lot of money. Next. So this is a famous example of Retnovir. So Retnovir is sold under the brand name of Norvir, which is a medication used for the treatment of HIV or AIDS. For normal formulation, form 1 was formed because, but however later the less soluble form 2 began to precipitate in the saturated solution in the semi-solid capsule formulation. So form 2, which was formed later, was less soluble in the solvent used in the formulation, which resulted in lower effectiveness in the patients. That means patients need to take extra dosage for the same effect for form 2 as compared to form 1. Then from then on, form 2 could be produced. So they had to rework on the suspension formulation, costing them a lot of money. So this case has been discussed in podcast here. Here is the link. It is interesting case study for polymorphism. So I would recommend to listen to this podcast. Next, the next question is how do we measure it? So the polymorph can be checked by Raman spectrometer, which can be easily integrated with the crystalline instrument. The Raman with crystalline allows you to real-time non-invasive tracking of the polymorphism, and there are various Raman spectrometer that can be seemingly integrated with the crystalline. Next, so this is an example of this is an example of polymorphic study of mefenamic acid with crystalline. So mefenamic acid is a drug used in the treatment of pain, mild pain, and it has 3 polymers, and form 1 being the thermodynamically most stable form. So in this study, form 2 was suspended in ethanol and stirred at 700 RPM using an overhead hooks impeller at 25 degrees Celsius in the crystalline instrument. Using the Raman spectroscopy, it was possible to track the rapid conversion of the mefenamic acid from form 2 to form 1, which is the stable form. Next. So similarly, this is another case study of carbamazepine, in which the treatment, which is used for the treatment of epilepsy and neuropathic pain and has different forms. So in this study, first the metastable form 2, which has needle-shaped crystals, were formed at higher supersaturation and then transformed into stable form 3, that has block-shaped crystals. Next. So these are the images of the carbamazepine taken with the crystalline instrument. Here we can see the carbamazepine nucleating as form 2 needle-shaped crystals, and then over the time, the solution-mediated polymorphic transformation happening, and the crystals transforms into a block-shaped crystals, that has stable form 3. Next. Coming to particle shape and particle size distribution. Next. So the particle size distribution has huge effect on the downstream processes and its efficiency. So these are the common steps followed in the downstream processes. First is filtration. A broader particle size distribution can result in poor filtration and trapping of impurities and solvent. Then milling. Milling is required to reduce the particle size and have uniformity in the particle size distribution. But during the crystallization, if you are able to achieve the desired particle size, then the milling process can be removed and it can be cost effective. Then further mixing is affected by the particle size distribution. A broader particle size distribution results in unequal and creates localized high concentration. And then the broader particle size distribution makes it difficult to standardize the amount of binder and excipients affecting the formulation of an API. Then for tableting, then different particle sizes have different compressions and breakage properties which affect the tableting. So therefore, it is very important to monitor and control the particle size and particle size distribution for the manufacturing of an API. Next. Apart from the particle size, so particle shape has significant influence on the downstream processes, specifically the filtration efficiency. So this is an example of how different particle habits can affect the filtration efficiency or performance. First example is the needle-shaped crystals with a lot of space in between, which can trap solvent because solvent cannot be easily drained out. Then the high residual solvent can result in impurity inclusion and slow filtration. The second is the filtration of polydispersed. The second figure is the filtration of polydispersed cubes. You can see it here. The packing space has reduced because of the small spaces in between or pockets. Trapping of solvent and impurities inclusion are possible. So this type of packing and particle shape results in moderate filtration efficiency. Then here is the third example of filtration of monodispersed cubes, showing high control of particle size distribution and represents good packaging. Because there are no solvent pockets, the chances of solvent trapping is also less and results in fast filtration. Therefore, to achieve high efficiency of downstream processes and desired API properties, it is important to monitor the particle shape and particle size distribution. Next, to monitor the particle size and particle shape, Crystalline is the instrument. So latest version of Crystalline instrument, Crystalline V2, has 8 optics where there is option of colored and mono cameras. Different camera magnification can be configured from 0.5, 1 time, 2 time, 4 times, and 5 times magnification, and the field of view changes accordingly. So this is an illustration or figure of a reactor inside an instrument. So there are 2 front light sources and 1 backlight source to increase the quality of the images taken. And then this is the camera and the transmissivity sensors for the turbidity measurement. Next. So this is a case study to see the effect of solvent composition on crystal habit. These are the images taken during the cooling crystallization experiment of ascorbic acid in water alcohol binary solvent system using the crystalline instrument. The effect of solvent composition of crystal habit can be seemingly noticed here. More cubic shaped crystals were formed in water, and with increase in alcohol composition, more elongated crystals were formed, resulting in rod-shaped crystals in methanol and in pure ethanol. So next, similarly, in water isopropanol solvent system, more elongated crystals were formed, and in pure isopropanol, cluster of needle-shaped crystals were formed. So these images were captured using the crystalline cameras. Next, so similarly, these are the different images taken with the crystalline camera showing different processes such as agglomeration, crystallization, forming, and oiling out. Next. These images shows different particle shapes. First is the elongated crystals, and the second are the cubic shaped crystals, then needles, then block shaped crystals. So these are just to show the different crystal shapes can be easily captured with the crystalline instrument. Next, so addition to just visualize the particles with cameras, the latest version of Crystalline comes with AI technology to detect and characterize these particle shape and particle size distribution. So AI is advanced technology which has the ability to detect the particles that it has seen before. So Crystalline gathers a set of images where particles are clearly marked, and this collection of labeled images form the basis for teaching the AI to spot particles in the new picture. Next, how it works. So how AI works. First, the input. Arina, you can click 2-3 times. First, the input. The process begins with providing the AI system with the images containing the particles. Then for the processing, the artificial neural network or the brain uses the deep learning technology with advanced technology with learned patterns from the pre-processed images to analyze the input images. It looks for specific features or characteristics that indicate the presence of the particles, and the particle separation will divide into different classification with matrix information. Next, after processing, the AI system provides an output that indicates the size and the shape of the detected particles. So this is the how the background of the crystalline instrument works with the AI technology. Next. So the particles are characterized into 6 different shapes. First is non-convex, which represents a cluster of needle-shaped crystals. Then quasi-equivalent, which means equal in all direction and represents the cubic-shaped crystals. Then needles, which are elongated crystals. Then platelet, which represents the plate-like crystals. Then sphere crystals, which are round-shaped crystals. Next, the size parameters that are characterized by the software are particle size, which is the equivalent sphere diameter, then contour parameter, which represents the parameter of the actual crystal, then contour area, which represents the actual area of the crystals, then ellipse shape factor, which is the major axis upon the minor axis, then convex hull parameter, which represents the outside of the crystals, but including the extra area as represented by the red line. Then the convex hull area, the same area, including the extra area with this red line. Next, further, the AI system gives information on particle size distribution with D10, D50, and D90 parameters. This is the verification done with the glass beads of hundreds of 100 micron size particles. Next, the instrument uses the most recent technology of AI to identify the crystals automatically, and with this technology, the AI is able to identify individual crystals pixel-wise. So finally, in the end, coming to the end of the webinar, I will share some examples to show different cases of images detected by the AI. Next, so detection of multiple habits. This is an example of carbamazepine. Here you can see that different crystal habits are identified as by different colors, needle-shaped crystals in red color and these cubic-shaped crystals in green color. Next, then overlapping particles can also be easily detected by the software. Here you can see that different needles which are overlapping are detected as separate crystals by the AI. Next, and these are the clear particles of, and here you can see that again the different particles are detected at different colors with different shades of the crystals. Coming to the conclusion, next. Coming to the conclusion, so crystallization is a commonly used process in industries for purification, but also controlling and influencing the particle properties that have significant impact on the downstream processes. Then Crystal 16 and Crystalline are versatile platforms that can provide vital information for designing a crystallization process at small milliliter scales. So, for example, solubility can be measured with the Crystal 16 and Crystalline instrument by the transmissivity technology and by capturing the images. Then nucleation can be automatically determined by the Crystal 16 instrument using the software by classical nucleation theory by measuring the induction time. Then polymorphic transformation can be tracked by integrating a Raman spectrometer with the Crystalline instrument. Then particle shape and particle size distribution can be tracked or identified or characterized by the Crystalline instrument specifically using the new AI software. Next, and with this, I would like to thank you all for taking your time out and attending this webinar. I am happy to answer your questions, and you can also write to my email ID, jyoti.yadav@Technobis Crystallization Systems, and also at info@crystallizationsystems.com. Thank you.
Thank you so much, Jyoti. That was very extensive description of some of the technologies available within Technobis Crystallization Systems. So with that, do we have any questions? Question has popped in.
Yes, I can see the question. So question is, how is the solubility measured using Crystal 16 comparable to gravimetric method? I think, yes, it is very much comparable. Even I can, there are few papers, even I also compared the solubility of ascorbic acid in water from using the Crystal 16 instrument and with the data available in the literature. So it was very much comparable. So yes, the data, if we get from the Crystal 16, is as accurate as gravimetrical method.
Oh, great. Amal, thank you.
So how many cameras are used for tracking images of crystals? So there are 8 positions for the cameras. So you can use 8 reactors at the same time. So, and with each reactor, there is 1 camera. I hope this is what you wanted to ask. Yes. Any challenges you face for Crystal 16, particularly for colored compounds? Yes, Crystal 16 for colored compounds, because it affects the transmissivity, so there could be some challenges. But it depends upon the color, how much color it is. If it is too dark, then then it becomes difficult. But if they are light color, light in yellow, light orange or whatever, brownish color or little dark, then still Crystal 16 transmissivity technology works, because there we have done some experiments for the to determine the solubility of compounds which are used for organic electronics. So those compounds are mostly colored. For those, Crystal 16 can work, but definitely if it is too dark, then the transmissivity technology is a bit doubtful. For that, as I recommended, you can use the crystalline instrument, where you can see the crystals appearing in the images. So from there, you can determine or select your clear and cloud points that will give you more accurate values of solubility for colored compounds. Only one side of crystal is captured with only one side of crystals is captured, which is fed to AI model. Only one side. Is it that means, is it not 3D structure, or I'm not exactly sure, because AI model, it works with the instrument. Means if the instrument is with the instrument, so it gives you the AI analysis in situ or real-time analysis. So so it is not fed to a model, so it will take the images which are taken are automatically analyzed by the AI software. If you have the AI software, if you have the AI in the instrument with the crystalline, then it will do the analysis real time. Then the next question is it possible to track the polymorphic transformation? Yes, it is possible to track the polymorphic transformation using a Raman spectrometer. You can integrate any Raman spectrometer with the crystalline instrument, then you can do the real-time polymorphic transformation of the crystals. Next question is, specifically in compounds like flufenamic acid that have multiple polymorphs. Yes, as I said, yes, you can do the polymorphic transformation by using a Raman spectrometer, which can be easily integrated with the crystalline instrument. Sorry. I hope I answered your compound. Sorry. Have you compared the particle size measurement results with FBROM, Blaze, or Recurrent? Not exactly. So because both technologies are different. FBRM works on different technology, and particle size measurement with the crystalline instrument is different technology. So it's difficult to compare, but yeah, so it's but we work in the future to standardize the particle size distribution as compared to, especially with the master sizer. So we can do that in future, but for now, because these are different technology, it's difficult to compare the particle size distribution. So there is another question by Amol. Can you compare with G3? I'm not sure what G3 is.
Hello, am I audible?
Yeah, you are audible.
Oh yeah, you are getting particle size from images. So is this like the morphology G3 also use the image technique? So it get the images of the crystal and then it gives the PSD. So I was just interested that if it is like the particle size distribution what you are getting is comparable with the image based technique.
Okay. Yes, I have not gone through this technology, and maybe I was not aware of this as well. But now you have shared, so this is good for us. We can do the comparison with this, because if this is the same technology of measuring the particle size with the images, then maybe we can discuss, and maybe I can do some comparison of the particle size with this with these technology as well. So
Okay.
that is good. Thank you for sharing this. Yes, if you want to write to us, you can write to me and to the crystallizationinfo@crystallizationsystems.com for any further questions. We will be happy to answer your questions.
Fantastic. Okay, we might leave it there for today. So thanks so much again, Jyoti, for your time and for sharing all of that information with the SSPC community. We really appreciate it.